4.30. Dynamics of infection rates of tsetse flies fed on trypanotolerant and trypanosusceptible cattle infected by T. congolense by Sophie Thévenot and Fabrice Sommet. Please show yourself.
Well, it seems you're still in the poster session. Shall we go on? Or? Yeah. Okay. So the third best poster, that will be what? The second of
That is post 4.26, being analysis of the distribution of tsetse flies in Masai Mara ecosystem, Kenya, by Kiteto Ndaburu.
Okay, so the second best poster
is poster 4.27, and that is enhancing and updating the National Atlas of Tsetse and Trypanosomiasis in Zimbabwe by Learn More Nyakupinda.
Is everybody there? No. Okay. Okay, we are maintaining the suspense until everybody is here. I'm not very good at keeping the floor. Worse. Okay, we have number 4 awardee there, Sophie and Fabrice. Sophie and Fabrice, show yourself. Yes, that's number 4. Okay, is everybody ready for the big announcement of the day? Yeah, finish with that, okay. Okay, and the big winner is?
Poster number 432, and that is the role of Stomoxis in... the role of Stomoxis in animal trypanosomiasis transmission dynamics by Julia Muita.
Now number 5, which is Mashlala Nkosi, if she's here. Ah, okay. So Moiti, you're gonna do the pitch for Mashlala. So now. On the poster. Okay, Moiti, I've just been told by our chairman that you talk too much, so you have 2 minutes.
So what happened here was we sampled the KwaZulu district, which is shown to be the area. So we have and then as we normally use, and we had to identify the two different species, and from there we had to do the parasitological identification where we have to do DNA extractions, PCR using ITS initially, and then from the one we have different PCR bands from the different species that we detected. We had to now do species-specific PCRs to then discriminate, okay, until we sequence them. And then on the tip there, we managed to detect a different Trypanosoma species. And so far in South Africa, Trypanosoma congolensis is still the most dominant Trypanosoma species circulating in our tsetse flies. And we are yet to sample more areas where now we are trying to identify more possible tsetse flies and also Trypanosoma parasites. And this is just a preliminary analysis, by the way. More experiments are yet to be conducted, and thank you.
Thank you, Moiti. So I will call now, I think it's Fabrice and or Sophie for a pitch.
Thank you very much. So you have already heard this morning Fabrice Homé. And this poster is linked with the presentation he showed you this morning about cattle trypanotolerance and interaction between different cattle breed, susceptibility or tolerant cattle, and hematological and biochemical variables. And we took the opportunity of this experiment to assess if tsetse flies that are fed on different cattle breeds could have different infection rates. There are only 2 experiments that were done more than 20 years ago to assess the effect of breed or species of animals on the infection rate in cattle. And so, can I have, yes. So we infected 3 cattle breeds, susceptible cattle like zebu and crossbred cattle between zebu and European breeds, and Daba cattle, who is tolerant to trypanosomosis. And we decided to feed tsetse flies from Glossina palpalis gambiensis at 5 points during the infection dynamics. So we infected cattle, and after we fed tsetse flies roughly around 20 days after cattle infection, 50 days, 80 days, 120 days, and finally 150 days after infection. So this experiment was done at CIRDES, who has the facilities to infect cattle and to rear tsetse flies. And after we assess first the survival of tsetse flies that were fed on the cattle at the 5 points during infection, and we assess the infection rate of tsetse flies by microscopic analysis and observation of gut and proboscis. And finally, I will go there to show you. So roughly, during the experiment, more than 2,000 tsetse flies were dissected by the entomologist of CIRDES, and they found more than 200 positive flies, so we have an infection rate in tsetse flies of roughly 10% during the experiment. But what we observe that we have main difference of dynamics, and the higher infection rate were found roughly at 50 days after infection, so at that point. And so there is a big effect of the date of feeding of tsetse flies on cattle on the infection rate in tsetse flies. But up to now, we haven't found an effect of cattle breeds on this infection rate. And so I really like to thank you, the CIRDES team,
because it was a tremendous work to rear these tsetse flies and to dissect and observe the gut and proboscis under microscopic. So thank you.
So now, okay, naomba Bwana Kiteto wakuje hapa.
Good afternoon, everyone. So I'm delighted to have been the third best. So now my poster is about spatial analysis of distribution of tsetse flies in Masai Mara ecosystem. This was my MSc thesis that I pursued at the Institute of Tropical Medicine in Antwerp, Belgium. So basically, Masai Mara ecosystem is an area that is bordering the Serengeti, so it's connected to the Serengeti National Park in Tanzania. So basically, the study involved deployment of traps. What I did is analysis of data. So during the monitoring, 185 traps were deployed in that area, both biconical traps and good traps. And the results were that presence of Glossina pallidipes, Glossina soenatoni, and Glossina brevipalpis. So the most predominant species was Glossina pallidipes. So what I did is spatial analysis to show the presence or absence of the tsetse flies and also abundance. Further, I did spatial analysis using SAT-scan to see the likelihood of presence of tsetse flies. So what I used is the predominant species, that is Glossina pallidipes. So this results here shows where the Glossina pallidipes were highly concentrated. And out of the 54 traps that had the presence, 30 were found in that, spatial cluster. So further, I proceeded to do multiple factor analysis using presence or absence as categorical variables and using bioclim data. Bioclim data 1 to 19 is related to precipitation and also temperature. So basically, I wanted to compare whether we can use multiple factor analysis or whether they are correlated with the spatial analysis. So the multiple factor analysis, I used factor min R, that is in R, and then went further to do hierarchical clustering of principal components. So the conclusion is that the results of the multiple factor analysis show that specific species are linked to specific traps that are deployed in the areas under certain climatic conditions which are temperature dependent, with seasonality having a negative effect on the catches. Thank you.
Okay, so to continue, the second prize, so learn more. You talk a lot as well, that I remember. So 2 minutes.
Thank you so much. I will not waste too much time and go back into the methodologies and stuff because most of it is the same. So first of all, I just want to acknowledge the combat project, as all of this work that we've been doing is feeding into our work package 3, that is for the Atlas enhancement, because for us we had already published our first Atlas in 2021. So after the first edition was made, there were some that we identified after doing our mapping. So those areas are the ones we are addressing now. The first edition was mainly based on tsetse traps, epsilon traps. And now in our enhancement, we want to try to factor in as well other entomological survey methods that we're using, especially the fly rounds. Then we also need to also factor in PCR results for validation, because when you look at the poster, you realize that there are some areas where we really have negatives in terms of there's no tsetse and no AAT presence. So those areas, we just need to do sort of like a validation so that we can clearly demonstrate that there is indeed freedom in those areas. So over and above that, the Atlas has been a really good tool for us in a way to say that we can clearly and easily identify priority areas in our country where we need to invest more in terms of control, invest more in terms of surveys. So we know where our gaps are, we know where our problems are. So what remains now is just for us to do the work. Then in terms of the flies, in terms of their abundance, in our first edition we had something like 38,000 records, and now we've only added 3 years and it's now 40,000 plus in terms of sites which have been sampled and the records that we now have. So for us, each record represents a monitoring that was done for each site, for each trap. And for our animal samples in terms of AAT, we did an additional 5,990 samples over the period of 3 years. And this was generated from 93 sites. That is our deep tanks, a select of the deep tanks that are part of the nationwide network of deep tanks that we have. So in short, this was the work that we are doing and where we intend to be or where we intend to go in the future. So we are still working on it. Expect more in the few years to come. Thank you.
Thank you, Learn More. Last but not least, Julia Muita. Okay, and very importantly, and it was not a criteria in choosing, we have a lady as a winner.
Okay, as I've been introduced, my name is Julia Muita, and I'm glad to be the winning award poster. And this is my work that I've been doing under the supervision of Dr. Merid and Dr. Masiga, who is here. So for introduction, we all know trypanosomiasis is a disease that is devastating to the livestock. And the objective of this work was really to see if the biting flies transmit trypanosomiasis, because we also find that there is trypanosomiasis outside the SSA belt areas. So for our methodology, we did both the molecular work and the experimental work, and so that to incriminate stomoxis in the trypanosomiasis transmission dynamics. First of all, we collected the trypanosomiasis from the field, and the first thing we did was the blood meal analysis, whereby we wanted to see which livestock at risk, and at the same time, we also wanted to see, do really stomoxis, is there a spillover from the wildlife to the livestock, because we know the pathogens and the livestock, the wildlife are the reservoirs of the pathogens, and we can you can see on my poster, those were the results, whereby stomoxis feed on various livestock and also various wildlife. And then we also did analysis molecular on the blood from the host animals, and at the same time we also did the pathogen diversity in stomoxis from the various regions where we collected the blood from the animals. And then further, we did the vector competence, whereby we saw that the stomoxis could transmit the trypanosomes in vivo, though the rate was not so high, but it still plays a role in the transmission of trypanosomiasis. Thank you.
you, Julia. So, just before closing, so first, the award will be a all expense paid trip to New York. No, will you will receive a certificate which is being printed and will be handed to you tomorrow. I'm just the messenger. Now, just a couple of remarks and maybe recommendations. So you may have seen that the 5 posters that were selected, I won't say they look alike, but they are, let's say, conventional poster. Well, usually if there is convention, there is a reason, and those posters had everything that was needed to have a clear, well understandable poster. And most importantly, who did it, where it was done, what was the aim of the poster, what was the conclusion, references, etc. Because we have seen posters without authors, we have seen posters we didn't know in which country the work was done. So that's not very attractive for someone to read it. I have a couple of recommendations. First, okay, that's a bilingual conference, but is it good? It's bad. I don't know. But if you want to reach a large audience, you better do your poster in English. It's pretty easy nowadays to do that. As much as talking English may be difficult for some, I know about that. But writing on a poster is not that complicated. Okay. Well, the other recommendation is what I commented. So maybe, Verl, you want to add something?
For the introduction, I saw some very long introductions, but without any objective or without any reason explaining why the work was done. So I think that is an important point of attention. Materials and methods should be clear, but not too long. And then also in your results, if you put figures, maps, and an explanation, be sure that they correspond and you don't mention names of places in your text which cannot be found back on the map, for example. For the conclusions, very important, you only make your conclusions based on the results you showed and not based on something that is not on the poster. And for the references, you can have a few references, but don't forget to refer to them in the poster, and also the references that are referred to should be in the reference list, which was not the case for some of the posters.
Thank you, Verl. So I think I will give back the floor to our chairman, as this poster session is over. And thank you to all, and congratulations to the winners.
You agree with me that there was good justice done on the posters. Again, to re-emphasize that the posters, when you're asked to prepare a poster, you should never see it as second class. The way I view it is that you have a chance to present all the information you want to present, which is different from when we have to make a presentation here for only 10 minutes and you have a chance for only 2 or 3 questions. In the poster, you are able to interact as much as possible with those who are interested, and you also have something that you can put on your wall. So it's a very, very important way of communicating our technical work. The judges, if I could call them now, the judges did a great job considering the time that they were given. So a round of applause to the judges, and thank you very much. Thank you. Thank you. I don't want to forget the rapporteur. She was not sitting up here, but Mary, you have also been recognized. Now we are getting into a very interesting session. A very interesting session because this insect that we see here is a very unique insect. I am not an expert in this area, but this is a guy who only feeds in blood. This is a guy who is a very poor producer. He reproduces very poorly. This is a guy who, an environment that he is in, if it is interrupted a little, he very quickly disappears, or she. Yet this guy has kept Africa hostage to both human and animal African trypanosomiasis. So what you are going to be listening to today is more about the sassafly and what is being done, what constraints they're bringing, and what approach is required to be undertaken to address this. And to take us through this, we have the moderator, Dr. Rajida Saini, if you could please come to the podium, and the rapporteur, Dr. Ajakaye, if you could please come to the podium. So I hand over to Dr. Saini to lead the process. Dr. Saini.
Good afternoon, everybody. Welcome to this last session of the conference, which will go on till tomorrow, on Glossina biology, control, and eradication. So I would like to first of all call our keynote speaker. I met John in 1983 or 84 when we had the ISCTRC meeting in Zimbabwe. So he comes with years of experience in sessile biology, especially ecology. If you look at any ecological papers or modeling, John Hargrove's name will be there. So without much ado, John, the floor is yours.
Thank you, Rajinder, for your kind remarks, and thank you very much for inviting me to talk to you this afternoon. Let's hope, Collins, it's working. That's great. So we're going to be talking today about how things have changed and how they're changing even now and how they will liable to change in the future. But let's just remind ourselves about the things that have stayed the same. One of the things is that tsetse flies are very slow breeders. As the previous speaker actually noted, they are very slow breeders. They only produce one offspring every 9 days, maybe every 12 days. That means that if you can maintain really quite a modest mortality among the adult female population, R0 in the terms of epidemiology goes below 1 and the population declines, and if you maintain that pressure, that population will go extinct with probability 1. And what you see in this graph here, coming from a paper that I wrote probably 20 years ago now, I hate to think how long ago it was, but even if you've got 0 pupil mortality in your population, if you can kill around about 4% of the adult females per day, you'll get elimination. And if David Rogers is right that the pupil mortality is more likely to be actually more around about 1%, then you only need to kill 3% per day and you'll be home and dry. This seems ridiculous, but in fact, when we actually, when Glenvale organized the first use in anger, as it were, of the odor-baited targets in the Reefer Triangle, what we expected to happen happened almost exactly. The other thing is, I'm afraid the top of the slide has been cut off, but the rubric at the top just says some things never change. One of the things that never changes is you never get your slides quite right. Anyway, as the gentleman said before, also, what we know is that tsetse feed only on vertebrate blood. So if there aren't any animals, there won't be any tsetse flies. And this is demonstrated in this slide. An experiment done even before I started working in tsetse in the 1960s, where they fenced an area, chased in northern Zimbabwe, chased out all of the elephants, and then proceeded to destroy all of the tsetse hosts, shoot them over a period of a number of years. It went from on the right-hand side, there's a log scale of the number of, no, that's a linear scale of the number of animals in the blue, which went down from 2,000 down to about 200. So they removed 90% of the animals. And as they did that, you see the red, these are the numbers of flies they caught on the fly round, which is on a log scale. And that went from, they were catching upwards towards 2,000, and it went down to the point where they were only catching 2 flies a day. In the black is basically just showing what was happening in a control area only 50 kilometers away. So no animals, no tsetse. And Bourne, Reed, and David Rogers and Wint basically pointed out that actually, and they were quite right, they wrote a book actually on autonomous tsetse control, pointing out if people move in, actually cut down all the trees, the animals will go anyway. You want this for farming, you want it for crop production. There's nothing for the tsetse to live on, and they go. And this autonomous tsetse control has been going on forever. And as they say, with continued human population growth and environmental change in the years to come, the further retreat of tsetse and elimination of wildlife can be anticipated. So in a lot of ways, we've developed new ways of doing things. We know that it's actually not that difficult to get rid of tsetse flies, and yet, of course, we are still struggling. So what are some of the difficulties? We can see what things are helping us, but what are some of the difficulties involved? Well, one of the things is that the tsetse flies do actually move. They don't move as rapidly as insects like locusts, but they move rapidly enough that actually they can reinvade an area that you clear, and they can do it surprisingly quickly. Again, this is a paper I published about 20 years ago, and basically I was asked by the FAO to do a modeling exercise. What happens? You take an area of 10,000 square kilometers in an extensive tsetse belt, and you get rid of them overnight. If you could just do a very good aerial spraying exercise, get rid of them overnight, how quickly will it be reinvaded? And the answer is ridiculously quickly. This down here on this alpha is basically equivalent to a fly which is taking a step length of about 1 kilometer a day, which is on the high side. But at that sort of time, what it basically is saying is within 600 days, the entire 10,000 square kilometers has been lost. If you actually say, well, that's rather a high value, if we take it down to if they were just doing about 400 meters a day, it doesn't make an awful lot of difference. It doubles the length of time, but basically you're very quickly back to square one. And you can see how in sort of early days of even the really quite big and nicely done tsetse control operations, they fell foul of this problem. So this is in Botswana, where aerial spraying was started in 1973. You can see there was a few hundred square kilometers done there, and then the next year there was some done further west. All right, you can see what happens. You've taken all the flies out of that area for the 1973 spraying block. While you're doing 1974, the flies are going back in over there. So you go, well, okay, they did it in 74, and then they did it in 75, 76, 78, and they kept on doing it, moving around. But even by 1981, they weren't really getting on top of the problem. But nothing ventured, nothing gained. They kept going until right the way through 1981 through 1985. And through again until 91, with bigger and bigger blocks overlapping in one thing or another. And when they actually had done all of that, they claimed in the 1990s that actually tsetse had been, I think they used the word eradicated. People were a bit floppy about using the word eliminated and eradicated. But the fact of the matter is that you apparently at that time you saw very, very few, you hardly saw a tsetse fly. And yet, a few years later, the flies were everywhere, which is exactly what we would expect given the modeling that had been done. So, but around about 2000, the Botswanians actually got serious. And they said, come on, we actually have to get on top of this. And what they did was to say, what we'll do is we'll take 2 blocks, each of them order 8,000 square kilometers. And we will spray them. We can't actually do all 16,000 in 1 year. But what we will do is we'll do this, the northern block. And before we start doing that, we will put in a band of odor-baited targets 10 kilometers wide. Now, I had actually done some modeling which basically showed if you use a 10-kilometer band of targets, there is almost no probability that, there's a 0 probability that flies will actually get across that. Of course, they get across there ultimately, but if you only wanted to keep them out for a year, you've got a pretty good chance with that density of targets of actually keeping them out. And so they did that, and the next year kept the targets in and came and sprayed the south. They did a little bit of an overlap in both of them. And long story short, there has not been a tsetse fly caught since, I forget where I put that, since 2005. Okay. So the point is, actually, tsetse control, and therefore the control of human and African trypanosomiasis, the elimination, it can actually happen. It can happen on if you've done it on a sufficient scale using appropriate technology. Okay. Now, this was actually done just at the end of the period when again, on the top, it basically talking about the regional tsetse control program in Zimbabwe, Zambia, Malawi, and Mozambique. And what had happened during the 10 years or thereabouts, in that decade of the 90s, was that there was a massive improvement in aerial spraying, the development of insecticide-treated targets, and probably most importantly in the long run, the discovery that you could actually use deltamethrin on cattle to get good kill rates of tsetse flies. And you can see the results of that. The tsetse veterinary department, extremely active in those days. They were looking at over 300 sites around the country, and were able to see which of those dip tanks and where they were testing the cattle, where they were red, indicated that you had positive for trips amongst your cattle, and where it was green, they were free. And what had happened is basically during the period between 1981, straight after independence in Zimbabwe, until 1997, there was basically almost a complete removal of animal African trypanosomiasis from Zimbabwe. And you can see this, right? This is the bars on the right, bars how many dip tanks were checked, and the dots are showing you how many cases. And you can see by the end of 1999, or the end of the 90s, there was basically, it was essentially animal African trypanosomiasis had been eliminated from Zimbabwe. Okay. Things have been difficult in the last 20 years, as we know, but nonetheless, the principle is maintained. You can actually do it. So what the RTTCP did actually find was that they had a huge amount of difficulty taking it the full step. What the intention was actually to eradicate the entire common fly belt of those 4 countries. And they came to the conclusion that, sorry, they didn't intend to actually do it. They intended to find out whether it was feasible. And their decision was that actually it wasn't feasible, in particular because different countries had different expectations from what they wanted. The Malawians basically wanted to just take care of tsetse around their game parks to stop the possibilities of human trypanosomiasis there. The Zambians had particular, what they wanted was to do with particular areas they wanted sprayed. The Mozambicans simply weren't in a position, they didn't have the finances to do anything. The Zimbabweans actually wanted to eliminate the flies from their country. So it was completely different. And they basically said this looks as if it's impractical. We can discuss that later furthermore. hence or otherwise, but actually more otherwise. Actually, at the end of the 90s, as we all know, there was actually a huge sea change. Donors withdrew en masse, and they cited a whole lot of things. One of the problems was, and I hold Rajendra Saini entirely responsible for this, is that we were too successful. People had, like Rajendra and also people in Zimbabwe, had managed to identify odors which could pull in huge numbers of flies. People had been using, getting insecticide-treated cattle and so on. And basically what the donors were saying were a number of things. You've developed all these nice tools. These should be used by the people, and they should be used by the people who own the stock. They're their animals. They should be paying for this. Now, you can say what you like about that. It's not something that finds great favor with me, but that basically was one of the things that they were talking about. What they also said was, we've been to places in Zimbabwe, even where the Tsetse are most serious, and when we ask the people there, what are their priorities, they say, our priorities are jobs, children's schooling, hospitals, clinics, etc., etc. Number 12 was Tsetse control. So if the people don't actually rate Tsetse control, why are we paying for Tsetse control? We should be paying for other things. And of course, this came along at the time when HIV was absolutely rampant. We tend to forget, because we've had COVID recently, that HIV was such an absolute disaster, and still is. And they also started being much more concerned with issues of looking at governance in Africa. So all of these things basically meant that suddenly, and still to this day, we really have very much less money spent by donors on Tsetse and trypanosomiasis control than we would hope, and indeed really should be actually spent. There we are. The result of this is basically what we have to face the fact is that we need to go for simpler, cheaper methods of disease control. But it's not bad. I don't think we need to get depressed about this, because actually there are good reasons, and people have been talking about these in the last few days, to think that, yeah, look, it's not all doom and gloom. This stuff can work. We can actually do this. And we've got some cause for optimism that the thing is workable. And one of the early indications of this was the SOS program in Uganda, where insecticide-treated cattle were used, and basically the frequency of our hats in the country went way down. And now later on, of course, tiny targets have been used all over the place, and you can see that when they were first used in Kenya, the blue shows what happened when tiny targets were deployed, massive reduction by 60 to 95% in terms of the numbers of flies, compared with in the gray with a control area where there was very little change. Now these are these pathetic little things. How can that be used for Tsetse control? Well, the fact of the matter is, they might only be pulling in 0.1% of the population per day. If you've got 1,000 times 0.1%, suddenly you're getting above the sort of level of 4% where you can actually bring the numbers down. More importantly, of course, the fact of the matter is that when it comes to human African trypanosomiasis, which people are very interested in this part of the world, and further north and west, of course, and quite rightly, you don't actually have to initially get rid of all the tsetse flies. You get it down to the point where you break transmission, and you get it down to the point where you actually get elimination of the disease as a public health problem, which has actually happened in these countries, in Benin, Cote d'Ivoire, Equatorial Guinea, Rwanda, Uganda, etc., etc. Now, do not think for 1 minute that I'm saying this is all down to the use of tiny targets. This has been a joint effort, WHO, chasing down people who are and treating people who are positive for trips and so on. But what people have come to accept is that the added use of these rather cheap and nasty tiny targets is actually a really good thing to add into the basket of techniques we've got. And the great thing is that they're twice as effective as traps, they're 10 times cheaper, they last longer, and they're easier to deploy. So there are lots of very good things to be said about those. I'm going to skip over that just right. Now, the rubric says, what do we need to be supporting? And basically what I think we need to say is, listen, in a sense, these tiny targets were whipped up by somebody almost on a back of the envelope. They've got to be placed, and we've heard people already talking about how, I think in Guinea, there have been modifications to the tiny targets, now using a cross-target, one thing or another. There got to be possibilities for making these cheap things better. We know we're going to have to use these, but can we actually make them better? And so what we need to be doing is we should be supporting and strengthening the use of these low-cost vector control methods. What we also need to know is where we should be focusing our disease and fly control efforts. And so one can laud the fact that the EU have actually, and I apologize to the other funders who are involved, but are actually, that they have launched the Atlas and combat projects. Now, these are important in themselves, but I think they're actually important also in the indication that maybe the donors are starting to think, you know, we actually should be getting back in there in some way or another. And what I hope is that what the EU have been doing there might actually be taken up by other donors. And if those other donors are actually going to be doing something, where could they actually usefully put their money? And what I think do point out is that the Atlas and Combat project, and particularly the Atlas project, is essentially basically telling you, it produces a map, which is telling you, at best, it will tell you where the flies actually are. And of course, if you do it again, you'll tell it. But what you would actually like to know, a map always tells you of where you are at the moment. It doesn't actually tell you where you're going to be tomorrow. And what we do need to do is actually to get a better understanding, improved understanding for various species about how they relate to changes in climate in particular, and how that's going to affect their distribution. So we need to actually understand that. Now, what this actually does is to point for an urgent need for actual field research. I know that there's some excellent work that will be being done with combat in Europe, and indeed there will be stuff in laboratories and one thing or another. What we don't see are actually well-focused field research stations in Africa. And I think that it's time that we actually need to think about those again. They have been in the past, and they have kind of fallen into desuetude. What I'm suggesting is, and it is a pity that that thing is not there, but basically that what we need to do is have centers of excellence in various bits of Africa where we can actually do that kind of research. And I'm in discussion with my co-authors, we basically, there seem to be three areas. Basically, Fuscipes for East Africa, Palpalis for Central West Africa, and Polydipes, Morstan's group for Southern Africa. Now, I absolutely do not feel that I'm in any position to suggest to people in anywhere north of the Zambezi, really, or north of Zambia, about where those a center of excellence for those, where those two might be put, and that is for discussion for those people. I do have a very long association. I worked for 30 of the happiest years of my life at Rukomachi Research Station, which sits in the Zambezi Valley. And what we have here is a research station which has gone through hard times, as we know in the last years, but to the great credit of Albert Mandurwa and his staff, they are actually raising that back up. There are staff working back down there. They've got cattle which it can be used for experimentation. They've got refuges, they've got trapping going on, and this is extremely important, and it deserves support. What we also have is we have got 60 years of continuous monitoring of what's been going on with the climate, and 60 years of almost continuous monitoring of what's been going on with the tsetse populations, and we can put those two together, which I'd like to talk about. This business of data actually raises a point. Before I do that. So the point is that we know, it's just, we all know that climate change is with us, and it's with us, and it is probably the biggest existential threat that we are facing. It knocks COVID into a cocked hat, it knocks AIDS into a cocked hat. Okay? And the interesting thing is that we are actually able, when we look back at our tsetse data, we saw this coming. We could see the effects on tsetse. In a sense, it's like a canary in the coal mine. It's telling you what's going to happen. And you could see this by going back and looking at these data which I got, these were traps that I had run to catch flies to do ovarian dissections. And the top graph is basically showing what happens with the tsetse flies, the numbers of flies caught per trap per day, done by months. And you can see on a log scale that basically it stayed fairly flat until about October 1992, and then the wheels fell off. And because it's on a log scale, you don't get the impression, but I can tell you that what happened was that within 20 days, the populations fell by 90%. And it fell by 90% because what happened was that the mean maximum temperature over the 20 days was actually above 38 degrees centigrade. And what happened thereafter was that there was a couple of years later, in 94, 95, and 96, there were hot years, and the population just sort of went down and down. So having been up catching 250 flies a day, you got down to the point where we were lucky to catch 4 flies a day. And this was actually really it looked as if this was very, very closely linked to what was happening with the temperature. And indeed, if you looked at the temperature, and as I say, we've got data for this entire 60 years, what you see is that if we take 1990 as a cutoff, to the right there, we've got, since 1992, the daily average 20-day running mean of temperature exceeded 32 degrees centigrade, which I think if those of you who know about tsetse flies know that above 32 degrees centigrade, things get extremely difficult for tsetse flies. The pupae just die. You can't actually get them to survive at that sort of level. And since 90, it has happened. It's gone. That running mean has exceeded 32 degrees centigrade 15 times. Prior to 1990, it only happened once. And on the bottom scale, of course, below 19, that happened 3, 7, 8 times prior to 1990, and only once there. So it's getting markedly, markedly hotter. Okay. So what happened was that I showed you the numbers of flies caught in traps after 2000, when basically everything kind of closed down because there was no more funding. But to the great credit of the government, they kept doing sampling. And what you saw, these are the red dots, which are showing the numbers of flies that you catch per afternoon on an ox. And again, it's on a log scale. So basically what they were required to do is to catch 50 fed flies for testing insecticides. And what happened was that prior to 1990, that's why there are no red dots, it was always very easy for them to catch 50 flies. Thereafter, suddenly they were coming back and saying, oh, sorry, could only catch 45, and then went down and down and down. And it went to a point where in 2016, to cut a long story short, it got down to the point where they weren't catching 50 per day, they were catching 1 fly every 10 days. If you actually do all the sums, you can actually work out that at the end of 2016, compared with the highest levels we had at Rukomachi, the population density had gone down by 5 orders of magnitude. And the squiggly line is basically the line coming out of the model that was fronted by Jennifer Lord, and it provides a pretty good idea of what's happening, of a fit to the data, where we're simply assuming that everything in what that happens with tsetse is actually just driven by temperature. The mortality, the birth rate, the rate at which the pupae develop, et cetera, et cetera. It seems to work extremely well. But we actually only used, okay, so then having looked at these, Joshua Longbottom at the Liverpool School actually said, well, suppose we use those data. What does it mean about what we might expect to happen elsewhere in Zimbabwe? Because what it basically says, if we had those kinds of temperatures, and we must expect that things will get worse in terms of the heat in the Zambezi Valley, it's going to become too hot for them down there. So what you might expect is the flies that happen to go up onto the escarpment will actually find things very much better there. And you might expect that, therefore, what you will find is an effective shift of the population from the Zambezi Valley up onto the Middleveld and Highveld. So the red here basically shows where we think there's very high probability of finding tsetse before. But what Joshua Longbottom found was basically when he used our sort of outputs, everything in blue basically says, from now on, we expect that this will become less favorable for tsetse. everything in red will become more favorable for tsetse. So it looks as if we might well be seeing a movement, an effective movement of flies out of the Zambezi Valley. The point is, though, this was using just a tiny bit of data. It was on one species, one sex, and just over a small number of years. Okay, we've got an awful lot more data than that. So, but you have to worry about what happens to data. And this is a picture sent to me by David Rogers, and he took it in 1988. And this is the archives for the data collected in from the 1920s onwards, and for heaven knows how long. And that's how it was stored. And I think you can have it in writing that the rate of the storage will not have improved. Okay, they will probably be will have been lost. Now, it's not that gentleman's fault. It's not the fault of the government of Tanzania. It's not the fault of Jackson, who was long dead. There was nobody there there was probably nowhere to store it in the first place. There was nobody to actually archive that data in the second. And that's a tragedy. If data are not properly archived and made available, they'll be lost. They'll be lost to everybody forever. Now, as I say, the modeling that's been done, which Joshua Longbottom used and which Jennifer Lord did, was based on one sex, one species, sampled over a limited period. But we've got a lot more data than this. Okay, these data look actually much better than you saw in the last tour. They're in nice boxes on shelves. They're in what used to be my office until I was kicked out as director. Thank God. But the point is, having them there is one thing, but actually if nobody knows what's in those boxes, then they might as well be burnt. And so there is a problem, and we need to actually, we need to get those data archived and put out there. And that's a difficult job. You have to have people that they've got to be annotated, not simply just put onto the computer, but you've got to tell people how they can use these data. And that's not an easy task, and it basically needs to be undertaken by people who actually know about the data. And so that is what we're trying to do, is get some support to actually do that. It's going to involve people like myself and Vale and Tor, but it's also going to require people to actually do, collecting all the data. Yes? Sorry? Wind up. Okay, 1 minute. Okay, I think we can just do that. So let me just, just the last two slides. What do we want to do in the future? Well, we'd like to as we say, extend the model to do from 1959 onwards, base the model on on meteorological conditions, and use the models to predict future distributions of tsetse and trips in Zimbabwe and elsewhere. And of crucial importance, I've talked about this. So I'm not going to stress that point again, but just to say what we have got. We've got ovarian dissections on over 180,000 females. Nutritional data, that's to say fat, residual dry weight, hemoglobin, etc. We've got TRIPS infection data on 50,000, and we've got photo images on over 100,000 tsetse wings with people who are interested in morphometrics. Okay, mark recapture data from 5 years of antelope study, and plenty of stuff there. So it's that there. And basically, so that's just the most important thing, getting back from looking at the actual data, is just remember, the first aim of what we do is basically to get rid of the disease. In theory, if you could get rid of the disease, there was no more disease, you wouldn't really care too much about the tsetse flies. We care about the tsetse flies because only we think if we don't remove them, the disease will come back. But the disease is what we are really interested in. So first, get rid of the disease, and then think about eliminating the tsetse. We know that that's going to be difficult, where we've got people where it goes across borders. And so approaches to tsetse elimination is going to be country-specific. But I would like to stress on behalf of Mr. Mandurwa, who actually points out that as far as Zimbabwe is concerned, tsetse elimination is their top priority in order to ensure the full realization of production potential of the smallholder farmer. And we certainly support him in that. And thank you very much for your attention. I'm sorry I've run over time, but I do want to thank very much the AU IBAR for providing financial assistance so I could come here. Thank you.
Thank you, John. I think we can keep the questions for later as we are running a bit late. Let me call on the next paper, 4.01 by Thierry Demus. Population genetics of Huskippies in southern Chad.
Thank you. So first, I would like to thank the organizers for this nice meeting, allowing us to share our results. So I'm Thierry Demus. I'm working in the Research Institute for Development in France, in Montpellier. And I'm going to talk to you today about our results on the population genetics of Glossina fuscipes from southern Chad. So we had the opportunity to sample tsetse flies in this area where there was two foci of human trypanosomiasis in Mandul and Maro. And we had sample of tsetse flies before control campaigns against the tsetse flies. Unfortunately, not at the same time for constraints, because of some constraints. So the first sample was in Mandul in 2013, then in Maro, a long time after, 22 generations, about 22 generations after. And then we had also two samples in Timberia Dokutu, where there is no sleeping sickness, but which allowed us to study the geographic effects on population genetics. So we studied microsatellites markers, and the main results. The first one I wanted to talk to you is some curiosity we observed about densities. So here you've got, you've got a nice correlation between the density of captured flies and the effective population density. So the effective population densities is about the density of fly that can give viable descent to the next generation. So this correlation is a good news because it means that both mean something that has something to do with biology. But what is curious here is that when we had very high densities, then the effective population density was much above the observed captured density. And the other correlation was between the sex ratio, which was strongly female biased for the less dense populations. So it was some kind of puzzling results. And the best explanation we found is that when densities are very high, it means that there is a lot of resources available at short distances. So tsetse flies do not need to move a lot. So they are not captured a lot by the trapping devices. And this concerns as well female and males, which would maybe explain why the captured flies are less abundant, apparently, than what is really in the population. And when the densities are very small, it would mean that resources are scattered in the environments, not easy to access to, so the flies need to travel a lot to get access to it. Then they are more vulnerable for the trapping devices. And this would affect more female than male because they need to feed much more to produce the larva. So besides this curiosity, as far as the population genetics is concerned and the population structure, what we could find thanks to the Dokutu Timberi samples is that we could estimate that the immigration was between 1 and 2 individuals per generation, every 6 months, sorry. So we extrapolated for Mandul, which are distant from Tiberi about 60 kilometers, as is Dokutu and Tiberi. We also noticed, because of the structure of our data, that the temporal structure was the main factor here. As you can see, the Mandul subsamples are all together and far from the others, genetically speaking. And the others, even the most remote one, are together because probably they are temporally much less distant. And then here we've got a result. Probably because it is on the border with the Central African Republic and probably receive a lot of immigrants from there. And then the most isolated spots, like Mandul, Timbuktu, are less genetically diverse. And we can also see that there is some overlapping, suggesting that some immigration can occur between these sites, but probably at a very low rate between these isolated spots. Within each focus, we noticed that dispersal is almost or totally free along the forest gallery. So it's here about 30 kilometers long and here about 25. Then I will skip that to save time, but we noticed a strong signature of a bottleneck of these populations. And I skip to the conclusions. So on average, we observed quite, on average, low levels of population densities and rather long distance dispersal. A very limited gene flow between the different zones, and in particular in Mandul, that appeared quite isolated. And the eradication here is because no tsetse flies can be observed anymore. So what we recommend is still to get, even at a very low level, some kind of surveillance just to check. It's better to be prepared than sorry. But achieved, there are still tsetse flies, and probably because there is no control on the other side of the border, and the focus is still receiving flies from there. So we suggest that the controls should still continue to protect human populations from infection by the trypanosomes, as we know that control, even if tsetse are not eliminated, control efficiently protects humans from being infected. And then I think, yes, we had a signature of the brutal decline. Probably in the 90s or later, meaning that before that, probably there was a huge population, a continuous population of tsetse flies that became subdivided in small pockets. So that's all. This, sorry. Can I? Okay, so something is missing in this slide. I'm sorry. This was published in the Peer Community Journal. You can see, if you want, that there is a poster describing the Peer Community in Consortium and the Peer Community Journal, the Peer Community Infections, which is a diamond open access way to publish. So it's free for authors and totally free for readers. And then we encourage you to submit your best papers to this PCI infections, or even apply to become a recommender, which is the same role as associate editor of a regular journal. So thank you for your attention.
Thank you very much. We move to the next paper by Kennedy on host preferences of Austini and Pelidipes in Kilifi. Kennedy, please.
Thank you, moderator. I'm Kennedy Ogola from Biotechnology Research Institute, CALRO, formerly known as GETRI. I'm going to share with you the work we did in this region, where we looked at blood meal patterns of two tsetse flies, that is Glossina osenai and Glossina pallidipes. So we have a big team. I'm Kennedy Ogola, like I said. and we all work under Dr. Paul Mireji. So I think some words are blocked there. But since Monday, I think we have heard a lot about SESE, AAT, and HAT, and the big menace they cause in this continent, especially sub-Saharan Africa. We also heard that there's some work going on towards development of vaccines. We can only hope that they'll be successful. But to date, we don't have vaccines for AAT or AHT in the market. Control efforts therefore rely on chemotherapy and vector control. There are several methods used under the vector control, but I'll focus on artificial-based technologies that rely on visual and order lures. So there are several targets and traps that have been developed that are usually used in these control methods that have, basically they have different variations of blue and black colorations depending on the species that you are targeting. And these targets and traps are usually deployed alongside the order, such as poker, fermented cow urine, and even acetone. So we know that poker was developed from preferred vertebrate hosts of the savanna species, mostly from buffalo and the cattle. So most savanna species of flies respond well to poker, but this particular one, the geoseni, does not. And to date, we don't have an effective attractant against that particular species, which hinders control efforts towards it. So we believe that identification of different host preferences between geoseni and G. pallidipes can establish additional host candidates that can be exploited to formulate new or to improve the existing attractants. And that was the objective of this study. So we carried out this study in 2 protected areas in this region. That is Shimba Hills National Reserve in Kwale County, where geoseni and G. pallidipes occur in sympatry. And also we did another study in Arabuko-Sokoke National Reserve in Kilifi County, which only has geoseni. So we believe that the population of geoseni in Kilifi and the one in Kwale are allopatric. So the 2 protected areas have rich wildlife diversity, as you can see, including buffalo, antelopes, bushbucks, and elephants. So what we basically did was to map the entire reserves for presence of tsetse flies in accessible areas. We then divided the accessible areas into blocks and deployed a series of goo cloth traps and sticky panels baited with poker and some with fermented cow urine. We also placed sticky panels placed around tree trunks and then collected the trapped flies every 24 hours. So this map shows where we first deployed the traps, the upper maps, and then the lower maps shows where we found fed flies. So we identified the trapped flies using taxonomic keys, then visually establishing their feeding status, and confirmed some by microscopic dissection. And we basically squashed the midguts on Watman filter papers, number 1. And there are some gut contents that we preserved whole in 70% ethanol. So the smears were air dried under shade and wrapped in grease-proof materials, and then stored in airtight desiccator. So we transported the samples to the lab, where we did the usual, did the genomic DNA extraction using a BioLine kit as recommended by the manufacturers. Then we employed PCR coupled to high-resolution melting, targeting the 16S ribosomal RNA gene product as previously described by Ouso. Then we purified the product and sent some of the applicants to INCABA for unidirectional Sanger sequencing. We got the results, we analyzed and aligned the sequences using bioedit, that version, and confirmed the vertebrate species by sequence alignment at 99% homology via basic alignment search tool. And then compared this against the National Center for Biotechnology Information and redundant nucleotide database. So the results, so it trapped the highs of over 10,000 flies, but only 398 G. pallidipes and 204 ausenine flies out of those had blood meals. And from those, we successfully extracted DNA from blood meals of 300 G. pallidipes and 157 G. ausenine. And in Kilifi, we collected a total of 122 G. ausenine and successfully extracted blood meal DNA from 30 G. ausenine, meaning that most of the flies were tenerol. When it comes to putative vertebrate hosts identified, we found about 6 species in both two sessile species. So we had Ernest bushbuck, African buffalo, common warthog, cattle, bush pig, and Sunni antelope. So in terms of differences in the two species, we recorded significantly more Ernest bushbuck and Sunni antelope blood meal in ausenine compared to G. pallidipes, and then recorded significantly more common warthog blood meals in G. pallidipes compared to G. ausenine. In Kilifi County, we also detected Sunni antelope, Ernest bushbuck, African buffalo, and cattle. And the frequency of the blood meals, most of them was from Sunni antelope, that is the 63.22, followed by Ernest bushbuck, buffalo, cattle, at those percentages. So discussion and conclusion, we note that no effective attractant exists against G. ausenine, which is hindering control programs against that particular species, and that identification of preferred G. ausenine host can inform formulation of attractants specific on the order profile of the host. So in this study, we established that G. ausenine predominantly feed on Sunni antelope and Ernest bushbuck, and we believe that the two vertebrates are viable candidates for bioprospecting for the attractants effective against G. ausenine. So I thank you for listening to me.
Thank you so much, Kennedy. Now we move to sexing of pupae by infrared imaging. Chantal, please.
Good afternoon, everybody. My name is Chantal de Beer, and I am from the Joint FAO-IAEA Center. And today I would like to show you a little bit about how to sex tzetzes in a new way. So first of all, so historically, if you look at the differences between males and females of insects, it's very easy to see. The males look different sometimes from the females. Their antennas look different. Their body shapes look different. And there is a segment at the end of the body that sometimes are different for males and females. Sorry. And this is also the case for tsetse. The 9th segment is modified in the males, so it's very easy in the adult phase to see the difference between males and females. But if we consider a control technique such as the SIT, which is the mass production of insects, and then the sterilization of radiation of these insects, the release of these insects in the field, that will then mate with the wild individuals, and there will be no offspring. Only having to know when the females and males looks different in the adult phase is not the best, because we would already want to know when we are producing these flies, if they are males and females. And we want this because in the Zanzibar SID program, they had to employ 21 technicians to separate 110 males per week. So it's very labor intensive if you only have to separate the flies one by one. We also want to know where are the males in the pupa. In the Senegal project, they wanted to irradiate the pupa as they didn't produce this in Senegal, and they wanted to ship it from other mass rearing facilities. So if you consider we would need an automatic sex separating system for pupa would be best for us. And we can consider the pupa. So there are sometimes slight differences in the size, but this is not always repeatable. So you cannot always use the size to separate the sexes. We can also look at there is a variation in the emergence time. So the females, they do emerge first, and the males after them. And you can find complete sexing of some species if you incubate them at a constant temperature of 26.5. But there are some difficulties here. So there's an issue with the incubation temperature. It has to be that is shown on the pupa. So let's consider the pupa again. Here is what it looks inside. But if you turn it around to the side, it looks like that, and then like that. So it's important that you look at the pupa from the right angle. Otherwise, you'll miss the black box. So you put the pupa in there, and then they go through there, and there's a little conveyor belt that moves them around so that you can take pictures of the pupa while they're moving. And this is what you see on the screen. So in that little square, we do this. So there is an algorithm that has these three questions. So in this square, how many of these pixels do you find any dark pixels? Is the answer is yes. Then for the next question is, how many dark pixels do you find in this square? And if that is yes to the threshold that you've set, it also asks the question, as the pupa go along, how many pictures have dark pixels? And if all the questions is yes, according to your thresholds that you set, the actuator goes on and the pupa is sorted into the melanization side. But it's very important, again, to consider that you need to, these pupa need to be developed, there needs to be synchronous collection of these pupa, and they need to be incubated at a specific constant temperature. So we did 3 assessments to validate this machine. So the first, what we wanted to know, these 3 thresholds, what is the best parameters to set, and when is the best time to sort our pupa. So you can see a male at the side. So what we did is we ran it through the machine at different ages, and we recorded the images. Then we found that for our information, there are many thresholds that you can use to be able to sort between the males and the females. So it will be important for your situation and your species, because all this is for Glossina palpalis gambiense. All the information that I'm going to say today is for this species. So different thresholds will be important to set for your situation, as well as the age of the fly will be dependent on the species. Then we wanted to know if we put the pupa through this machine, if they will be damaged. So what we did is we did 2 assessments. So we put them directly onto this conveyor belt in the first assessment, and then we fed them 3 times through this conveyor belt. So we put them in, they will roll, and they will fall into the unmelanization side, and then those pupa we will sort again, and another way, another again. And then we also had a new feeder that goes into the system. We put the pupa inside the feeder and see if these rollers would mechanically damage them. When the pupa are sorted, we take the pupa. The pupa are then placed in these little black tubes. They are left to emerge, and it's coated with talcum powder, so they cannot crawl out, they have to fly out. And in this way, we can assess if they are damaged. So if you look at the flight rate and the sorting accuracy of the two assessments, so the first one is when we didn't sort our pupa. Then we sorted them once, twice, or 3 times. And the last 3 parts is no sorting again, and then when they go through the roller and when they do not go through the roller. And there was no significant difference in the way they are able to fly out of this tube if they are sorted or non-sorted. If you look at the loss of females, this is the amount of females that are still remaining in the pupa when you already sorted. If you sort them twice and 3 times, you reduce the number of this contamination in the pupa. And if you sort them with the feeder, there's no difference between the contamination with the GAF. Then we also wanted to know, okay, now we sort 200 fly pupa, but what happens if we sort 13,000? So we, in collaboration with the Slovak Academy of Sciences, we had this setup of 2 machines, one in our laboratory and one in their laboratory. And we saw if it can be repeatable, if you can sort larger patches of flies to the same accuracy. So IPCL is us, and SAS is in Slovakia. And there was quite a little bit of differences. So we sorted between 200 and 2,500 pupa per day. And they sorted between 3,000 and 13,000 pupa per day. We collected our pupa twice a day, so in the morning a batch and at 3 a batch, and they collected the pupa only once a day. We sorted at a different age range, and they sorted at a slightly different age range. And remember our threshold there, so threshold 3, we didn't adjust it. We had the same amount of threshold all the time for all sortings, but they adjusted it every time that they sorted. So this is the results. So at IBCL, in 10 months, we sorted 280 sortings, and we sorted 229,000. And for SAS, they sorted 132,000. And for the male recovery, so this is the amount of males that you get out of the sample that you sort, they had a much better male recovery than us. So SAS had much more than us. And the reason for this is that we did not adjust our thresholds, but they did. When it comes to female contamination, we almost had the same amount. So in conclusion, the mean recovery can be improved by adjusting your metallization parameters. We can sort up to 36 pupa per minute. That means around 2,000 per hour. For a colony of 100,000 production of females, you can that has a production of 0.6, which is sort of the average. 4 hours, you can sort all the pupa that is produced by that colony. So if you consider manual sorting, one very experienced technician can sort approximately 1,000 flies per hour using a chiller. But with the machine, you can sort approximately 4,200. The next step to consider is how to use these sorting machines to incorporate into colony production to make it a little bit more easier to do. When I still have a little bit of time left, right, I will talk about my unit that I come from, right? So I will talk a little bit about the insect-based control section and the insect-based control laboratory and our functions. So you know, I'm from the Joint FAO division, and I want to give some information about what we do as an insectary. So we provide these functions to our member states. We provide services and advice to our member states. We foster information exchange. We promote capacity building and technology transfer, and we conduct need-driven applied research. So just a little bit of information about providing services to our member states. So at our laboratory, we have 7 species and 8 strains of tsetse. So if you require any tsetse that is on this list, please consider that our colonies are not mass rearing colonies, but small colonies. You can ask us to get samples, and we will send you pupa if you request. So this is a service that we provide. We also do a radiation dosimetry service for insect mass rearing facilities. So if you want to do insect radiation and you want to make sure that the dose that you're giving to your insects is actually that dose, we do dosimetry for you. Sorry, please also have a look at these books on the screen. You can find copies of them on our website. Also of these manuals. So if you contact to the website, there will be a few manuals that we have developed that are free of charge that you can have access to. So please go have a look at the website. So we also do need-driven research, and this is done at our laboratory, and you can we have a newsletter every 6 months that you can have a look. It's also on our website, so please have a look at these. But we also provide capacity building, so we also do 6 months and 1 year fellowships. We also have PhD mentorships and internships. So please have a look at our website. Many times this is advertised, and we would like to train young people from the region in these type of things. We also have CRPs, and currently we are having a CRP on the colony management and insect pest rearing for SIT application. This will go up into this year, and then the next upcoming CRP, please keep your eyes open, will be on SIT population genetics and distribution modeling for SIT applications. And we are very interested in the riverine species as well as brevet pulpus and osteni. We also help our member states to ask for extra budgetary funds, so you know that we are the agency and we can have access to funding like the peaceful use of irradiation. And we have done so in the past, especially for the Senokal project, that we do help them with additional funding for their projects. Thank you.
Thank you so much, Intel, and thank you very much for the offer of supplies and capacity building. In fact, IEA, we need to thank them for undertaking capacity building in so many countries for so many years. So can we move to the next paper now? Radiation of Palpellis gambiense, Somalia.
The slide will be in English, but the speech will be done on French.
Our presentation is just to remind you this work was done perhaps 50 years, which defined the dose of irradiation in sterile mile for Glossina. They're the same data which have been used up to now. For us, we have a station in Bobo in Burkina Faso, which is an insectarium for African West, which should produce male flies. So we need to have something on quality. As you do know, Caballol has said it's on the basis of the success of eradication in Zanzibar that was born the PATEC program. That means that for this success is the technique of the sterile male which was used. We have to produce a quantity of sterile males, irradiate them, and then you release them in nature. Then there will be no more reproduction. As I was saying, for this technique to be effective, the males which will be produced, the main element is sterilization of the males. If the males are badly sterilized, the program will not succeed. If you sterilize the male with a very high dose of irradiation, they will be sterile but will not be competitive on the ground. If the males, if the dose of irradiation is high, as the males are not very competitive on the ground, your program will not work, or else you use more money to attain the objectives. So as I was saying, you know that in Burkina Faso there was a program of Burkina Faso in the year 1980s in the focus of Iradui, and for Palpellis gambiense, it was defined as 100 degrees for eradication of the males. It is since 1976 that dose was defined, and it has been used as the reference dose, even if there is also some evolution of tools which are used for irradiation. So for us, we are using other equipments for this irradiation, and it was necessary to revisit these dose to see if we should keep the same dose of 110. As I was saying, here we use, for example, an irradiator, a cobalt irradiator, with two sources for sterilizing the male, and so we send this to Senegal, and that's the machine used for sterilization. To verify the optimal dose, we use 7 groups of male people, and these groups were irradiated at different doses, from 110 to 170, and the 8th group of males, which we are going to use in Benin, the control group, which were not irradiated. Now, the males of 7 days were used with female fertile female, and they were monitored. We were able to measure the rate of fertility so as to see the status of the females, and also to determine we should also evaluate the performance of these males, because it is the males who are irradiated and will be used on the. When we used a technique called Fulken's so as to assess the performance of the males, there were 3 groups of females, males irradiated at 120, another group at 140, and another group which was the control group. Now, we then release 30 females, and 10 minutes afterwards we also release 30 male. So 90 males which were going to compete for the 30 females. This was repeated several times, and at the end we were able to assess the performance of each group of males. And the results which we obtained showed, for example, that the fertility of this group was reducing with the increase of the dose. And this figure from the table which you can see there, it allowed us to determine the rate of sterilization induced. The results which were presented here by Chantal showed that in order to use this technology of irradiation, the irradiated male should be transfer sterility to about 95%. Our results showed here that the weakest dose which would allow to attain this level of sterilization was 120 degree. So from these results, we concluded that our optimal dose was 120. It was 110, but we reached at 120. And the same dose was used for other species which obtained superior doses. When we did the dissection to see the status of the reproduction of females after 2 months of monitoring, for females which mated with the males, the uterus contained either an egg or a larva, or it was perhaps empty. If the uterus was empty, it was a female which had just perhaps laid an egg. With the status of women who have mated with that if in the uterus there was 1 egg, that would mean 1 egg. You can see the picture I'm showing there. It is an egg. a female which mated with a sterile male, that shows the difference that we saw in the status of reproduction. To assess the performance of the male, we were able to show that there was the same performance with person irradiated at 120 degrees. There was no significant radiation difference. It's just too fast. So here, as I said, we didn't find any significant difference in terms of performance between the males of the control group and the males who were irradiated with the 120 optimal dose of 120 degrees. So here we found that the dose of 120 degrees did not affect the performance of the male which was sterilized with this dose. And if the male is sterilized with this dose, it could compete on the ground with the other male, and it will. To conclude, just a reminder, our objective was to review the optimal dose for sterilizing Gotsina pallis. In our conditions, we saw that the optimal dose is 120 degrees, which allow it to sterilize the results of this study confirms the quality of the males who were irradiated with this dose and which are sent to Senegal. If the results are satisfactory on the ground, then the eradication is attained, and then Senegal will confirm eradication in the zone of and confirm the quality of the males we sent to Senegal. Thank you.
We move to the last paper before the break. Rosemary, reinfestation of tsetse populations along the Uganda-Kenyan border.
Which one? I am Rosemary Bateta from Kenya Agricultural and Livestock Research Organization. I'm going to give a presentation on work that we have done on finding sources of reinfestation. That is by looking at the spatial genetic connectivity of Glossina fuscipes tsetse fly population along the Uganda and Kenya shores of Lake Victoria. As a way of background, we all know that Glossina fuscipes is a vector of human and animal African trypanosomiasis, which causes a huge economic loss in sub-Saharan Africa. As the earlier speakers have said, there are limitations in control methods. That is, we don't have vaccines, and also the available drugs have side effects, and also they are expensive, like in cattle, and also they are difficult to administer. Those are the human drugs. So the only viable way of controlling the trypanosomiasis disease is by using vector control. In this region, the Lake Shore region where we did our study, previous control efforts were done in the early 2000s, that is by PATEC and FITCA. And there was a reduction of tsetse population by 75 to 90% of the populations reduced. But after that, there was a reemergence of the flies in this region. So the question is, where are these flies coming from? So there are genetic studies that have been carried out on GFF in this region in previous studies, but these studies were carried out on small spatial scales. Those are scattered scales, but in our study, we wanted to look at a bigger spatial scale and with an attempt of trying to find out where are these flies coming from to reinfest these controlled areas. So this is a map showing the areas where the samples that were used in this study were collected. They were collected along the shores of Lake Victoria in Kenya and Uganda. We have 14 sites in Uganda and 6 sites in Kenya. And these samples here, they were collected by Iseni et al. 2012 that he used in his study. Then we added this one. So what we did, we used the 853 samples, extracted DNA. The new samples were 166, and genotyped the samples at 14 microsatellite loci. We determined the linkage disequilibrium, deviation from Hardy-Weinberg, did a genetic diversity, checked for the population structure, effective population size, checked for bottleneck, and isolation by distance. The information from this study is supposed to guide vector control planning and implementation. So after we did the genotyping, we moved forward, and from the results, we found that there were no linked loci, and also there was no deviation from Hardy-Weinberg equilibrium. So we went ahead and determined the genetic diversity of our population by determining the observed heterozygosities in the sampling sites, and also the expected heterozygosity, allelic richness, and the inbreeding coefficient. And when we look at this table, the HO, most of the figures, HO is less than HE, an indication that there is inbreeding within the sites, and this is also confirmed by the FIS, significant FIS, that is the inbreeding coefficient values. So after that, we went ahead to determine the genetic structure, of the population. So what we did, we used structure to determine the K values, and we found our best K value to be 6. And we used Bayesian clustering to assign individuals in the regions where they cluster. So in the 6 clusters. So we had the 6 structures, and when you look at this figure here, all the 853 samples are represented here. They are represented by bars. You may not be able to see them. And each color shows the 6 regions. So we have the yellow color for region 1, blue color for region 2. They have the pink for region 3, orange for region 4, green for region 5, and purple for region 6. But when you see the different regions, we have the mix of colors, which means an individual is at mix. That is, it has genes at least from the different populations. So from this, we conclude that the populations are admixed, an indication of gene flow. And the high levels of admixture is in region 3, 4, and 5. So we also went ahead to use a discriminant analysis of principal components to confirm our structuring in the previous slide. So we found out that there are still the 6 regions that are present in our population. And we also conducted population differentiation by finding out the FST. The data for this estimate is huge. That is why I could not project it here. But we found that there are low levels of differentiation among sampling sites within regions and among regions, suggesting high level of gene flow and interbreeding among many of the sites. So we also estimated the demographic parameters for each of the regions 1 to 6. So we estimated the inbreeding coefficient among the 6 sites. We also estimated the effective population size and checked for bottleneck. So from here, we found significant inbreeding within sites, and most of these sites are islands. So on an island, we expect a high level of inbreeding, and also there's a reduction in effective population size in region 5. But when you use the mode shift test in bottleneck, it revealed an absence of a population bottleneck in the recent past. So the reduction in the effective population size could just be because of the control effort that were done in the early 2000s, but they didn't cause a bottleneck, although the effective population size was reduced. So we went ahead to do migration analysis, and when we checked the migration of the flies in these sites, we found that there's a bidirectional migration of flies. When we look at these regions, we see this movement from one region to another, and the highest occurring between region 4 and 5. So then this answers our question, where is the reinfestation coming from? Is because of this migration that we are seeing here. And therefore, when we are doing our control, when we are planning our control programs, we have to be careful, because if, for example, we take region 2 and only implement control effort here, there are chances that it will be reinfested from the neighboring regions. So conclusions and recommendations. One is that there's an ongoing gene flow among the identified clustered sites. There's high genetic connectivity among the regions, and therefore there's need for sustained surveillance to reduce the potential of reinfestation in SESE-controlled areas. Also, we need continuous monitoring and control of SESE fly. For example, use of insecticides should target routes of human movements such as boats and ferries. From the map I showed you, the regions, most of them are islands, and you might be wondering why the flies are moving across the waters to the next region. There are boats and ferries that people use there for transport, so they could also be contributing to the migration. And the last one is that the two countries, that is Kenya and Uganda, should consider joint cross-border SESE control program. Thank you.
Thank you so much, Rosemary. Ladies and gentlemen, I think we can have a 20-minute break for tea, coffee. We are cutting it by 10 minutes, so you don't drink too much tea, because there's a cocktail at 6. So 20 minutes break, we come back, and then we finish the last few papers and have a good discussion. Thank you so much.